section, we're going to talk about how all of these organelles within the eukaryotic cell specifically work together to coordinate cellular function. So what I'm hoping that we'll get at the end of this section is an understanding of the membrane system within a eukaryotic cell and how that works to move materials from one part of the cell all the way out of the cell if necessary. To identify the function of the different parts of the endomembrane system, explain how proteins that are secreted from the cell are produced, describe the cytoskeleton, and explain how cells in tissues are held together, and then finally synthesize all of this information to really be able to talk about how a cell can function. So the information in this section is really going to be specific for eukaryotic cells. And while not all eukaryotic cells are going to do every single part, when you're talking about a multicellular organism, because the majority of the cells do these things, the multicellular organism will function. So something like a red blood cell, because it lacks a nucleus, is not going to be doing protein synthesis, and so it won't be doing all of these different things. But because other cells within the human body are, it kind of makes up for that. So these are generalized eukaryotic cell functions. They can't be applied to every single eukaryotic cell, and so that's one thing I want to caution you against when you're talking about really anything, but you try not to deal in absolutes. So usually if I say something like always or never, I'm trying to trick you, because almost nothing is an always or never. Never say never. So for the most part, these rules are applicable to the majority of cells, but just like if I said all eukaryotes have a nucleus, or all eukaryotic cells have a nucleus, you should be able to tell me one cell type for which that's an exception. That's what cells don't have a nucleus. So if I asked you, all eukaryotic cells have a nucleus, true or false? False. I lied. If I say most, then it's true. So most eukaryotic cells are going to have what we call an endomembrane system. So basically they have a series of membranous structures within the cell itself, and for the most part the function of these membrane systems is the transport of mostly proteins, but other substances as well. So within the cell, the major components of the membrane system include the nuclear membrane, which we know is contiguous with the endoplasmic reticulum. Then there's a separate membranous structure called the Golgi apparatus, which is not attached to the endoplasmic reticulum, nor is it attached to the final membrane that makes up the endomembrane system, the plasma membrane. So we'll talk about not really the nuclear membrane, simply because the nuclear membrane is mostly included because it's attached to the endoplasmic reticulum, but we'll talk in some detail beginning with the endoplasmic reticulum. So like I said in the previous set of slides and also just now, the nuclear envelope or the nuclear membrane is continuous, meaning attached to the endoplasmic reticulum. So basically a part of the nuclear envelope kind of blebs out. Bleb is a scientific word, by the way. I wish I was joking, it really is. And forms the endoplasmic reticulum. And when we talk about the endoplasmic reticulum, we can further subdivide it into what we call smooth ER and rough ER. Rough ER is called rough because it is studded with these ribosomes, so these little dots are supposed to represent ribosomes. Smooth ER lacks the ribosomes, so that's why it's called smooth. On this electron micrograph, they're trying to highlight over here, you've got endoplasmic reticulum, that's associated with ribosomes, so this will be rough. And over here on the left-hand side, the endoplasmic reticulum is not associated with ribosomes, so it will be smooth. The ER kind of folds in on itself, and those folds are called cisternae. And therefore, it also has these, you can basically have an internal part of the ER. It's sort of like a big balloon that's been all folded in on itself. So the internal part of the ER is called the lumen. So it's the space inside of these membranes. So even though smooth and rough ER are endoplasmic reticulum, they do have slightly different functions and roles within a cell. The smooth ER is one of the major sites of lipid synthesis within a cell. It's also a very important location for carbohydrate metabolism, detoxification of things that might have gotten into the cell, and the storage of ions, particularly calcium. The rough ER, as I mentioned, is called rough because it's studded with ribosomes. And those ribosomes are therefore the site of protein synthesis. So proteins are synthesized in the rough ER. Because proteins are synthesized there, they can also be modified there. So they can have things like carbohydrates or lipids attached to them. And parts of the membrane of the cell can also be synthesized in the rough ER. So that's the endoplasmic reticulum. So we've got our nucleus with our nuclear envelope, which is attached to the endoplasmic reticulum. We have two types of endoplasmic reticulum, smooth and rough. And if we continue moving closer and closer to the outside of the cell, we then have a structure called the Golgi apparatus. So the Golgi apparatus is basically this kind of clump of little membrane-bound structures that aren't really attached to one another. So you see it's kind of this discontinuous, right, these discontinuous little membranes that aren't necessarily attached exactly like the ER is. So there will be one side of the Golgi that faces the endoplasmic reticulum. So if we were, so we're in a cell, here's our nucleus, and then, you know, I'm not an artist, I'm a biologist. So we'll pretend this is, so here's my nucleus, here's my ER, and then this is my Golgi. So part of the Golgi faces here towards the ER. This part of the Golgi that faces the ER is called the cis face of the Golgi, or sometimes called, they show it here, the receiving side of the Golgi. And what's happening here in this gap is that products being synthesized in the ER are really being shipped in vesicles from the ER to the Golgi. So that's why it's the receiving end. And then what happens is, so let's say, you know, the protein was made here, and the protein is received at this end, then it's handed off, and then it's handed off, and then it's handed off, and then it needs to go somewhere else, so it leaves the Golgi. This is called the trans face of the Golgi, or the shipping side. Within the Golgi, not only is it functioning as this kind of receiver and shipper section, you can have additional modification of proteins. Sometimes that modification can be attaching a signal that says where the product is supposed to go. And the vesicles can go to other parts of the cell, or they can leave and go to the plasma membrane. So if we really think about what's going on inside of a cell as a type of shipping process, it would basically be like I don't know, you made cookies, and you're going to send somebody some cookies because you're really nice. So you made the cookies at your house, which is the ER. And then you take those cookies to the post office. And for me, I live closest to that Fig Garden branch of the post office. So I drive to Fig Garden, I drop off the cookies I'm going to send to my brother in San Diego. So that Fig Garden post office is the CIS space. It's closest to my house. And then the Fig Garden post office, it gets picked up and taken to a different post office, maybe the major Fresno post office. Then it gets taken to a different post office, maybe San Diego, because that's where my brother lives. And then it leaves the San Diego post office, and along the way, they might add another label that says it has to go here. They add a little barcode that say it has to go here. And then it leaves the San Diego post office, so it leaves the post office system and gets delivered to my brother's house. So that's what's really going on here. It's a one-way, right now we're talking about a one-way transport from where a protein or a lipid or something was synthesized in the endoplasmic reticulum, delivered via a vesicle or a little blebbing of a membrane to one part of the Golgi, the cis part, the closest part, transported through the Golgi to the trans part, and then it's shipped out to its final destination. So within the Golgi, again, there can be some modifications to proteins, additional tags that might say a protein goes here or there, and you can even get the synthesis of some polysaccharides. But the Golgi is like the post office. You've released control, and you're fingers crossed, hoping the cookies get there before they get back. So when something leaves the Golgi, it can go other places. So here they're showing you an example of an intestinal cell. So again, the majority of eukaryotic cells, even within the human body, will have these major functions or these major structures, but how they're organized might be slightly different. So for example, they're showing you an intestinal cell. You've got the Golgi right here. It's kind of hard to see in the micrograph, but then you have all of these vesicles that were shipped out through the Golgi, and these vesicles all contain mucin, which is basically the mucus that's going to line your intestinal tract. So they'll be shipped from the Golgi and eventually go out of the cell. So not all are going to have big mucin-containing vesicles, but that's one modification of this system. So here they're talking about a protein that's made inside of your cell and gets secreted and goes somewhere else. And so the example that they're giving you is insulin. So not all cells make insulin, only a certain type of cell in the pancreas. But insulin is a really important protein. But it functions in different parts of the body. So it has to be made in the cell and then exported. So they give you the steps here. We're in the ER. We make the protein. So that's what they're trying to show here. The ribosome is attached to the membrane of the ER. There will be a pore in the ER membrane that allows the protein to go into the ER lumen. Inside of the ER, that's where the protein gets folded. Because remember, three-dimensional shape of a protein is critical to its function. So it will get folded, exported, and sent to the Golgi. In the Golgi, it will be further modified and packaged into a vesicle. And then what will happen in this case is, so let's follow insulin. Let's say it was made here. So we've got our insulin. It gets transported to the Golgi, processed out, and so we still have insulin inside of our vesicle right here. So what generally happens in a case like this is that if we zoom in, here's our vesicle containing insulin, and here's our plasma membrane. These vesicles will fuse, and that will allow it to open up. So here's our insulin, here's our plasma membrane, and then the insulin can leave this cell, enter the circulatory system, and do what it needs to do. But why we say that the ER can contribute to membrane synthesis is that the vesicle that was made in the ER and secreted through the Golgi fused with the plasma membrane, and so now you have an additional part of the plasma membrane. So proteins can sometimes be secreted. Lots of proteins might stay within the plasma membrane and become embedded within the plasma membrane. But proteins can also leave the Golgi and go to other parts of the cell wherever they're needed.
Is the protein infiltrated form in the lumen?
Yes.
Then modified after entering?
So it'll actually go to quaternary form because it has two peptides that come together. But yes, it will usually have its final three-dimensional structure within the lumen. What we're talking about with further modification would be perhaps the addition of a small molecule that says where it should go, like a signaling molecule, or even something as simple as the addition of a phosphate group, like a phosphorylation. Yeah. But in general, what we want within the lumen of the ER is appropriate folding of the protein, because in the ER is a great place to check if the protein synthesis didn't go well, or the folding didn't go well. We'll destroy the protein right then and recycle those amino acids instead of have a misfolded or inappropriate insulin secreted. That's a very good question, though. So for the most part, yes, the majority of three-dimensional structure formation occurs within the lumen. There are proteins in here called chaperones within the lumen of the ER that help with folding. Okay, so any questions about that continuous membrane shipping system? So nuclear envelope, which is continuous with the ER. The ER will ship things to the cis space of the Golgi, which will then ship them out of the trans space, and they can go to places like the plasma membrane for final secretion. So another feature of a eukaryotic cell will be the cytoskeleton. So when we think of a human skeleton, we think of all of the bones that give us muscle attachments, the ability to walk upright, protection of our organs like our lungs, our heart, our brain. Cells also have a skeleton. And so the cytoskeleton, or the cellular skeleton, is important not only for movement of things like chromosomes, but also for anchoring of organelles, anchoring of nucleic acids, and allow some measure of support for a cell that doesn't have a cell wall. Cells that have a cell wall get the majority of their support from the cell wall, though they will have cytoskeletal components as well. We have three types of cytoskeletal fiber. We call them microtubules, microfilaments, and intermediate filaments. So microtubules are what we'll talk about first. They're made of tubulin dimers, alpha and beta tubulin. Microfilaments are made of actin. Intermediate filaments can be made of other things. Here they're illustrating keratin. And so when we're talking about microtubules, microfilaments, and intermediate filaments, we're talking about the size. So a microtubule is actually biggest. A microfilament is smallest, right, in terms of the diameter. And then an intermediate filament is intermediate in size between a microfilament and a microtubule. So a microtubule is a hollow rod. That's what they're trying to illustrate here with this ball and stick model. It's made of a dimer of tubulin, an alpha and a beta subunit. And they are in these alternating rings of alpha and beta. It's 25 nanometers in diameter, so this is 25 nanometers across. And microtubules play a lot of important roles within the cell. They help to maintain cell shape. They help in cell motility. Some of your cells do move. Other cells, like amoeba, they are unicellular. They don't have flagella. They don't have cilia. They move by modifying their cytoskeleton, and they drag themselves along by moving their cytoskeleton. So they can be important in motility. Chromosome movement in cell division. When you talk a lot about mitosis, what you see is, we duplicate the chromosomes and we line them all up in the center of the cell. You've probably seen those images before. And then we pull them apart so that every new cell gets a full complement of chromosomes. Microtubules are important in the lining up and pulling apart. And movement of organelles, which again can be important in cellular division because each new cell needs to have a full component of organelles in order to be functional. So microtubules also, as they're serving for organelle movement, they almost function as like an expressway within the cell. So molecules can move along microtubules and organelles can move along microtubules to move throughout a cell. They're capable of deforming and reforming fairly quickly, which is why when we talk about things like amoeba that use cytoskeletal components to move, what an amoeba does is it pushes out a projection of its plasma membrane. We call it a pseudopod, a false foot. So you have an amoeba, it pushes out part of its plasma membrane, and then it pulls itself along. And it pushes out plasma membrane and pulls itself along. And so the fact that these microtubules are able to quickly depolymerize and repolymerize is what allows those amoeba to move fairly quickly. They're probably faster than bacteria. Microfilaments are made of actin. It's 2 strands of actin wound together. It's very narrow compared to a microtubule, only 7 nanometers in diameter. These are also important in the maintenance of cell shape, changing cell shape, muscle contraction. So actin and myosin are 2 important proteins that allow for muscle contraction. And so the way that muscle contraction works is you basically have these different kinds of fibers that slide along one another and then go back, and slide and go back. And so that's muscle contraction and relaxation. So that requires actin and ATP. That's why when an animal dies, it eventually goes into rigor because you need to have ATP for those muscles, the muscle fibers to slide. When you're no longer generating ATP, the muscle fibers can't slide, so you can't move the muscle. Rigor generally breaks because the muscles have decomposed. So then it doesn't matter, they're falling apart, so it doesn't matter that they can't use ATP anymore. But so that actin and that sliding motion is really important for muscle contraction. So if you can't do that, you can't contract the muscles. They use it for cytoplasmic streaming or the movement of cytoplasm in plant cells, also cell motility, also cell division in animal cells. So a lot of these filaments and tubules can work together. These cytoskeletal components can work together to do these processes. The final cytoskeletal component are intermediate filaments. Intermediate filaments can be made of different kinds of components, whereas microtubules are always made of tubulin. Microfilaments are always made of actin. Intermediate filaments can be made of different proteins. For example, they can be made of keratin, They're anywhere from 8 to 12 nanometers in diameter. They're coiled up in cables, so they can help maintain cell shape. They can help anchor the nucleus and other organelles. They can also help form the nuclear lamina, which is basically like the nuclear cytoskeleton. So even the nucleus has a skeletal kind of component that helps it hold together. And so having these cytoskeletal components is critical for a lot of different functions, even when we're talking about cells in isolation. So a cell in isolation needs to be able to anchor its organelles. It needs to be able to divide. It needs to be able to do these things. But when we're talking about especially multicellular organisms, cells have to be able to work together and coordinate their efforts. In a unicellular organism, they can coordinate if they want. Things like bacteria do it all the time. They communicate, they coordinate their efforts. A lot of unicellular eukaryotes do it too, but at the end of the day, each cell of a unicellular organism is looking out for itself. So if the neighbors are bringing it down, it's going to abandon ship and go do what it needs to do. In your body, if one of your skin cells decides, screw these guys, it's just not going to happen, right? The body falls apart when a cell decides to do its own thing. Usually when a cell decides to do its own thing, we call it cancer. You have to coordinate your efforts in order to make the multicellular organism function. When we have similar types of cells working together, we generally call that a tissue. When you layer multiple tissues together, you can call that an organ usually. When multiple organs work together, it's an organ system. Multiple organ systems is an organism. But in order for those cells to work together, for all of the cells of your epidermis to form your epidermis, they have to be able to communicate with one another. They have to know, for example, when to stop growing. Because if they don't, that's cancer. Uncontrolled cell growth is cancer. They have to know when to grow. If we just take an example of your epidermis, if you get a hole in your epidermis, your cells need to know, oh, there's a hole, we should probably fill that up. The cells in your intestines need to know, oh, it's time to absorb nutrients. We should absorb more water. We should absorb less water. The cells within your nervous system have to be able to transmit impulses. Oh, that's really hot. I'll do it anyway. So it's really important that cells be able to communicate with one another. There are lots of different mechanisms that cells can use for communication. We'll talk about a couple of them. The first is that not all cells are going to be so densely packed that there's no space between them. So sometimes there are spaces between cells, or like spaces between different layers of tissue, like your epidermis and your dermis and your skin. And we call that space outside of a cell the extracellular space or the extracellular matrix. And even though it's a space, it's not empty. There are lots of important activities that occur in these extracellular matrices. Even your bone, which you think of as being very solid and supportive, has lots of gaps. And so these extracellular matrices allow for communication, they allow for some kind of structure formation, so they're very, very important. And one of the most important and abundant proteins in the extracellular matrix is collagen. So collagen can give support and flexibility to your skin, right? That's why they always try to sell, like once you hit a certain age, they're like, oh, you should buy collagen if you're going to get wrinkles. I'm like, well, I'm going to get wrinkles anyway, even if I don't buy collagen, so. But it provides support and flexibility to, for example, your skin. So as you get older, perhaps you produce less collagen. You don't do Botox, so you'll eventually develop wrinkles. Your skin is not as supple. So if you, again, look at the skin of somebody who's 80 or 90 versus the skin of somebody who's 8 or 9. But the extracellular matrix can also allow for communication between cells. So how do we get communication between cells? Sometimes we get the secretion of proteins, and sometimes we have cell-cell junctions. So in plant cells, we have a specific structure called a plasmodesma. And when you have multiple, they're called plasmodesmata. So what happens with the plasmodesmata is these are really, plant cells are really rigid because of that cellulose cell wall. So what will happen is where plant cells touch, there will be a gap or a channel that forms between the two plant cells. And that is, in one of them is called a plasmodesma, multiple are called plasmodesmata. And so this allows the cytoplasm to basically flow from one cell to another. That cytoplasmic streaming in plant cells. So this, again, can allow materials, let's say sugars or fluids, to be transported throughout the entire plant. But this is also how plants can communicate with each other. And again, for a long time we think of plants as they just sit there and don't really do much. But plants have a lot of functions that we still are learning about. One is if a plant is being eaten, it can send a signal to the rest of the plant, hey, I'm being eaten, we're being eaten, we're probably going to die if we don't do something, and so they can upregulate the production of toxic compounds or compounds that make the plant taste bad. And so they can communicate these things through plasmodesmata. I'm being eaten. They can detect, if you ever have a bunch of bananas, you put it in a bag and they all go bad pretty quickly. When one banana goes bad, it produces compounds that are secreted. The other plants can detect it and they start to write them too. So plants can communicate often through their plasmodesmata. Now, when we talk about animal cells, we don't have plasmodesmata because we don't have cell walls. So what we have in animal cells instead are what we call tight junctions. And that can be a watertight seal. Not all tissues are going to have this watertight seal, but some of them are. Things like your intestines have a watertight seal. Your skin has a watertight seal. And so that's what they're trying to show you here, is you do have some space, and then you'll have a tight junction or a seal. Space and a seal. It's like little brackets kind of studded throughout the membranes. And this is mediated by different kinds of proteins. I'm not going to ask you the names of proteins, but tight junctions are proteins that hold two cells together and form a watertight seal. And again, this is pretty important, and these are mostly found in cells of the intestinal tract, where you don't want things like intestinal bacteria to be able to free float into your bloodstream. You also don't want blood to be free floating out into your intestines. You don't want to be leaking fluids through your skin. You don't want blood to be leaking out of your blood vessels. So in those types of tissues, blood vessels, intestinal lining, skin, tight junctions are going to be more common. We also can have intracellular junctions found in animal cells called desmosomes. These are only found, again, in animal cells, and they use the example of a spot weld. I'm not into welding, so I'm like, I don't really know what a spot weld is. But they're basically in other tissues like the heart, in muscles. Instead of a tight junction, these are what allow the arrangement of the cells into sheets of tissue that can then, these sheets can then be layered together. And so that's what they're trying to show here. is a single cell right here on this side and a single cell on this side. And they're being held together by these proteins called cadherins. So it's not like a tight junction, it's not a waterproof seal, but it's holding, if you pictured additional cells coming off here and additional cells coming off here, if you picture what a muscle looks like, right, you can see those striations, like you can see layers that are held together, and that's the desmosome. So it holds especially epithelial cells together. And these are connected to intermediate filaments. So the cadherins that hang out of the cell are connected to the inside of the cell through intermediate filaments. We do in animal cells also have a structure that's a lot like the plasmodesmata. So that plasmodesmata that basically acts as a channel connecting one plant cell to another. We have channels that can connect one animal cell to another, and they're called gap junctions. Again, we can use them to transport ions, transport nutrients, transport other substances like hormones that would allow cells to communicate. And so while the function is the same, like these openings that allow cells to exchange cytoplasmic material, the structure of a gap junction and a plasmodesma are different, made of different proteins, have a little bit of a different organization, but at the end of the day, they have a very similar function. So the example, I guess, that's given in the notes on your slides is through heart muscles. So the electrical impulses can travel through the gap junctions to allow the heart muscles to contract, like to beat together. Okay, so the take home of this section is, one, we have this membranous network that connects our nuclear envelope to our ER. The ER can then transport things to the Golgi, which can then transport things to the plasma membrane or other parts of the cell. So you should know those parts and what they do. Proteins that are destined for secretion, the example that we talked about was insulin, will be produced at the rough ER in the ribosome. They'll then be folded and modified within the ribosome before shipping to the Golgi, where they can be further modified, before being shipped to the plasma membrane, where they'd be secreted. The cytoskeleton, unlike our bones, is a lot more dynamic and always changing. Every time the cell divides, it has to rearrange its cytoskeleton. If we're talking about something like an amoeba that relies on movement of the cytoskeleton, it's moving literally thousands of times a second. So these cytoskeletal components can polymerize and depolymerize very, very rapidly. We talked about 3 different types of cytoskeletal fibers: microtubules, microfilaments, and intermediate filaments. For the most part, a lot of them can play overlapping roles in terms of providing cell structure, cell shape, organelle placement. And then there are junctions that can hold cells together, and they all have different functions as well. So in plant cells, we have those plasmodesmata, which act like little gaps that allow cytoplasmic contents to go from one plant cell to the other. Then we have tight junctions in animal cells, or in some animal tissues, that provide that watertight seal. We have the gap junctions, which are a lot like the plasmodesmata, in that we allow for the transport of cytoplasmic contents from one cell to another. And then we also have our plasmodesmata, our tight junctions, and our desmosomes, which again, not so much a watertight seal, but like it's a spot weld. It holds like layers of tissue together, for example, in muscles like the heart. Then the gap junctions, which can also be found in the heart and allow for the transmission of electrical impulses. And finally, when we're talking about multicellular organisms, the organelles fit the function of that particular cell. So in a cell that has to do a lot of secretion, like we saw that example of the intestinal cell that has to secrete a lot of mucus, we had a pretty decent sized Golgi. We had all of those vesicles of mucin. Some cells may not need to make a lot of proteins or secrete a lot of proteins, like a red blood cell. So it won't have that membranous system like another cell would. Cells of the epidermis that make up your skin might have a lot of tight junctions because it needs to be more watertight. Whereas cells in the muscles that need to be more flexible and need to be able to move around may have more of the desmosomes, or cells of the heart, which need to work in tandem, have a lot of gap junctions. So the organelles and structures within a cell will really fit the function of that cell and tissue. So any questions on cells working together? Okay, so I want to stop here for today. We'll pick up tomorrow. If you looked at Canvas, you saw that I only put up 7, 8, 10, 11, and 12. 9 is something that we're going to work through together. So I'm not putting it up for you because we're going to work through it all together tomorrow. If you have not had your topic approved, please do that today. I've approved everything I've seen. I've either approved or not approved, so everything that was posted as of last night was commented on. So if you have not yet proposed a topic, you haven't got a response. If you have proposed a topic, you have got a response. So please check and make sure that it was the response you wanted. And I will give you back all 3 of your exams tomorrow as well.